Hi!
We're 10th graders Anya Pillay
And Asil Ali from Sage Hill School. This is our bacterial project,
RapiDegrade! Our project started by developing our scientific question, which asked, how does inserting DNA from Bacillus Megaterium into E. Coli K-12 improve the efficiency of plastic degradation by bacteria?
Part 1 of our study. Once our 2 bacteria brought from Carolina biological supply arrived, we placed them onto petri dishes filled with nutri-agar, which we made by mixing 10 grams of agar, 10 grams of dextrose, and 500 milliliters of distilled water. We then incubated them for 24 hours and started isolating the DNA from the Bacillus Megaterium. To do this, we used a process called heat lysis. We put a microtube which contained distilled water and the Bacillus bacteria into water baths at 65 degrees Celsius for 5 minutes. This temperature causes the cell walls to break apart, but to prevent too much damage, we put the tube into an ice bath for another 5 minutes. To force the remaining unwanted debris, such as lipids and proteins, to separate from the DNA solution, we pour our microtubule in a centrifuge for 10 minutes. The Bacillus DNA was then ready to be transferred into the E. Coli K-12.
Our second step was the transfer process. To do this, we first combined calcium chloride and E. coli K-12 from the incubated petri dishes. Next, we took a loopful of our Bacillus Megaterium DNA solution from part 1 and mixed it into the microtube. It was then time for heat shock. We put the microtube into a water bath at 40 degrees Celsius for 5 minutes, as this time the E. coli was more heat sensitive. We then transferred it to an ice bath for another 5 minutes. This process was repeated 3 times to ensure the Bacillus Megaterium DNA was transferred into the E. coli K-12 cells. Afterwards, we streaked the transformed E. coli K-12 solution onto nutrient agar plates and placed it into the incubator at 37 degrees Celsius overnight.
Our final step of the experiment was to set up the testing tanks. We filled 2 tanks with metal-rich soil at 37 degrees Celsius, put some PET plastic samples in each. In the first tank, we streaked the transformed E. coli K-12 onto the plastic, and we did the same for the second tank, with the original E. coli K-12. This creates a control variable to compare the modified bacteria against.
Overall, we came to the conclusion that the bacteria needed a little more time to notice a more significant difference. However, based on the trends in the data we observed, we can expect that the modified bacteria can degrade the plastic more efficiently, which is determined by the change in weight of the plastic. We would also like to continue this project and transfer DNA from Bacillus Megaterium to other fast-producing bacteria to see which combination is most time-efficient and most productive. This project tests a key concept in reducing the amount of plastic waste generated by humans.
Plastic degradation through bacteria is a viable strategy to effectively dispose of plastic and can lead to making our world a better place. By harnessing the power of bacteria, we can get one step closer to saving our world. Thank you for listening to our project, RapiDegrade!